Our speaker tonight has a very interesting subject. She has been studying the origins and evolution of life. And I have a quote. She is interested in how and why evidence of life in building blocks are preserved and what often isn't. And she has done field work with pre-Cambrian rocks in Brazil, Namibia, and the Southwest United States to develop her understanding of fossilized life from the right period. She received her undergraduate degree in Earth and Planetary Science and Astrophysics from Harvard. She has a master's degree in planetary science from Caltech, and she holds the National Science Foundation Graduate Research Fellowship. And for outreach, she works with the Pasadena Unified School District, developing elementary level science criteria. And she's quite an accomplished artist as well. And she is the recipient of a 2020 award from Caltech for her contributions to educational outreach. So with that brief introduction, let me turn the Zoom over to Cecilia Williams, who will explain about life on other worlds and what it would look like when we get there, when she gets there. Okay.
Thanks so much, Kim. That was a really sweet introduction and a really awesome news bulletin for here's all the things going on and all the major things going on in space sciences right now. So thank you for that. That was really cool. So as Ken mentioned, and as I'll re-explain now, I am not an astronomer. I understand that my my primary research topics are not necessarily the familiar fare for this particular group, but I did start my journey toward becoming a professional scientist as an astrophysicist. My undergraduate degrees are in astrophysics and planetary science. And though it may not seem that way from looking at my CV, I feel that my trajectory of interests has been pretty consistent since the very beginning for me. Since I was very small, I have been very interested in the origins and evolution of life in the universe. And I credit my parents largely for this. My parents are huge science fiction and fantasy fans, and my dad is a writer who writes about unusual alien civilizations, and and that always stuck with me. I wanted to know why is life the way that it is? Why is it located where it is in the universe? And how would we go about not just detecting it, but learning things about it, learning about the range of lifestyles available in the universe, the range of possible habitable environments, and how do we go about a comparative study of life as we know it and life as we can only imagine it. So I'm going to go ahead and share my slides here. I'm not going to be showing you guys any data today. This is all very conceptual. And I believe that we'll hold questions to the end, just to make sure that we get through things and for the ease of Zoom logistics. So bear with me just a moment while I share my screen. I started this talk, I've given this talk several times at different levels of technical complexity and to audiences with different kinds of backgrounds, but I think that the sort of universal appeal of the work that I do, no matter if your background is in is in astrophysics, or if it's in sociology, or if it's just, in the human condition. I think everyone at some point has wondered to themselves, are we, as in living things on the planet Earth, alone in the universe? What is it that makes us unique? What is it that makes us not unique? What is our relationship to the context of the whole? And ultimately, like, what is life? I think this is something that everyone in every field ends up asking themselves at some point. And for astronomers, the question is particularly poignant because so much of astronomy, so much of space exploration is motivated by the search for life. And if you're going to look for something, it helps to know what you're looking for and have an expansive enough definition of what you're looking for that you're being a good scientist and not necessarily biasing your expectations toward or biasing your findings toward your expectations from previous experience. So I drew this little cartoon, a kid asking their parents, "Mama, what is life?" as they look up what Douglas Adams described as the fantastic jewelry of the night. And I imagine the parent answering, "Well, baby, you'll know it when you see it." And the reason that I kind of start with this exchange is because for all of the work that scientists and engineers put into exploring the mystery of life in the universe, rarely is a lot of time given to the problem of what is life in the first place. What is it that we're looking for when we look for life in the universe, existentially, not even just analytically? And the best answer that researchers have come up with in the past several hundred years that they've been wondering this question is, you'll know it when you see it. And this is a bit of a glib, sort of tongue-in-cheek answer, but it really is one of the best answers that we have, because ultimately, what is life is sort of a semantic question. It's a question of language and a question of scope. And so, but it is one that still needs to be answered in order to do the research. So for the rest of this talk, I'm going to sort of step through our best attempt as scientists of defining the problem of life in the universe. And then I'll talk about, once we have an established sufficiently broad and sufficiently specific definition for life, I'll talk through some of the ways that scientists in different fields go about trying to find it and describe it and define it in ever more detail. So some of you might recognize the uniforms in this particular screen cap. This is a screen capture from the film Europa Report that came out some years ago now. Europa Report is one of my favorite science fiction films. I think it's one of the few science fiction films that starts to get close to what it's like to do field work, at least at the beginning. Things aren't very glamorous. You're sitting there looking at a bunch of sensors. You're stomping around a kind of barren landscape, in this case, the icy moon of Europa. In this case, the researchers in the film are looking to characterize the surface and subsurface of Europa. They're looking to understand, is there life here? And if I recall correctly, the way that they examine whether or not there's life there is to take samples of the ice, to take samples of the regolith on the moon's surface, and put it in a little machine and see if it comes up red or green. It's like, is it life? Is it not life? And wouldn't that be nice if we had a tool that could distinguish between those things so definitively. But Europa Report is just a slightly more recent and example of science fiction trying to sort of tackle this problem. An older and more familiar one for many of us is Star Trek, the original series, and beyond, or perhaps Jodie Foster and Contact. All of these stories, you see scientists with different areas of expertise, earth scientists, astronomers, linguists, specifically radio astronomers versus infrared astronomers and things, all trying to look for life signs, either on other worlds or life signs that might be emitted from other worlds and somehow received in the vicinity of our own planet. So as fanciful as some of these depictions of the search for life are, they're not so far afield from what we actually do as scientists looking for life. When we're trying to distinguish between something non-living and something living, rather than something dead and something alive, we step through certain criteria, and we use a wide range of analytical techniques. So the question of what is life boils down to can we define the difference between what is living and what is non-living? So most of us would agree, just instinctively, innately, we feel in our bones that rocks and water and the atmosphere, clouds, weather, all of these things are sort of non-living elements of our environment. Whereas microorganisms, bacteria, single-celled protists, up to animals, plants, fungi, we agree that these things are living. And even though the living and non-living are often intimately mixed and intimately related to one another, we're pretty confident in our ability to just innately know what is living and what is non-living. If we break down that sort of innate instinct, and go back to basics, when you hear scientists talk about the origins of life, the first spark of life, they're usually talking about the moment that you went from just a soup of chemicals floating around, dissolved in Earth's early oceans, the moment you go from that to having a distinct organism. And so in order to go from having an amorphous soup into a distinct organism, most scientists agree that you need some sort of medium through which these chemicals can interact with each other, and water is a very convenient medium for a range of chemical and physical reasons. Water is a polar molecule, meaning that it's got slightly negatively charged bits, slightly positively charged bits. It's very good at dissolving a number of substances for this reason. So it's possible to intimately mix other polar molecules into water. It's possible for water to help them dissociate and help them reassociate. So water is very important. We also generally agree that it's important to have rock or sediment. It's important to have some source of a range of different chemicals inside of the water. We agree that if you've just got water on its own, that doesn't really constitute life. You need something floating around in the water. And so rock, sediment, this is the source of a lot of those, of all of those chemicals that are dissolved into the water. I say some and all and waffle kind of between them because we're not even bothering with the atmosphere at this point. But just a soup of random chemicals floating in water over a mostly rocky planet, that doesn't really constitute life on its own either. There's some additional requirements that we innately feel are have to be met before we can say that we found life somewhere. So it turns out that the type of chemicals, the type of molecules that you have floating around in the water, those really matter, the definition of life. So most of us are familiar with terms organic versus inorganic. Lots of different kinds of chemicals exist out there. Organic versus inorganic, that's just one way of categorizing them. Organic molecules have a lot of carbon, hydrogen, oxygen, and may also have nitrogen and sulfur and phosphorus built into their structures as well. Organic molecules are able to achieve a high level of complexity and a high level of diversity of form that inorganic molecules aren't necessarily able to do. So carbon, hydrogen, oxygen are very good at making these sort of complex shapes and scaffolds. And it's through these shapes that most scientists agree that not just interesting chemistry, interesting reactions between different molecules, but reactions that are selective and directed, that's where they can start to happen. If you have a molecule with a very complex and structure, that molecule is probably only going to be able to react with other molecules under very specific conditions. And those other molecules have to be a very specific shape in order to sort of fit in with them and exchange the right number of electrons in order to create something new. So organic molecules seem a pretty important ingredient for life. We need that diversity of form. We need that complexity of form. But as members of the San Diego astronomy community, you already know that having a soup of organic molecules floating around, so organic molecules floating around in water over a rocky surface, that alone still isn't enough to say that we have life going on. Saturn's very famous, very hazy moon, Titan, is nothing but organic soup. Its atmosphere, its oceans are full of organic molecules reacting with one another. And yet, when the Cassini-Huygens mission did its flyby and dropped its Huygens probe and beamed back all the spectacular images of the moon's surface, we didn't really see anything that we would recognize innately as life. There's something still missing for us there. So next big step, that transition from a non-living world to a living world, is the idea of compartmentalization. Life as we understand it has to be enclosed or separated from its environment somehow. There has to be in order for something to be alive, we feel that it has to be distinct from the surrounding environment. Not that it's not interacting with its surrounding environment, but it has to sort of maintain some of its own identity separate from its habitat. And yet, in order to maintain this separate identity, that maybe counterintuitively means that it needs to be able to exchange. It needs to be able to respond to what's happening in the environment and change its internal conditions so that it can remain separate. So it doesn't just fritter away to entropy. So the way that life as we know it has accomplished this is through the use of membranes or walls that are semi-permeable, meaning that they allow certain materials at certain times to pass across the membrane, and in doing so, allow the chemistry inside of the membrane to maintain some sort of stability. And so many of you may be familiar with the term homeostasis. Homeostasis is the word that we use to describe this act of maintaining internal stability and maintaining an identity separate from the environment. But a bunch of organic molecules sort of enclosed in a membrane or a cell, maintaining homeostasis with the environment, we're approaching we're approaching something closer to a definition of life that we recognize, but we're not quite there. A burlap sack of dirt that you can shake some of the dirt out of if you move it enough. That's not a living thing as we recognize it, and yet it meets all of the criterion that we've established thus far. So it turns out that it's not just about having molecules reacting with each other inside of a membrane. It's not just about having organic molecules reacting with each other inside of a membrane. It's about what organic molecules are actually there and what kinds of interactions they're having with each other. So living things, as we recognize them, have to perform what we call a metabolism. This means that certain chemical reactions occur inside of that space that is distinct from its environment. Call it a cell for sake of argument. And that inside of that cell, these reactions have to occur that are releasing energy that can in turn be used to facilitate other reactions that are energy consumptive. So inside of a living thing, you have chemistry going on that wouldn't otherwise occur just spontaneously in the environment. You're transferring the energy of position from one state to another in service of building and maintaining this cell. So the coupling of energy producing to energy consuming reactions, that too is essential to our definition of life. Now, past that, there are some additional requirements, some additional criterion that not all scientists agree on, but I personally think are pretty essential, and that's the concept of self-perpetuation or replication. So in a more complete definition of life isn't just trying to maintain one state, but perpetuate that state and replicate it, create more units in a state similar to the starting state. So this idea of self-perpetuation replication is kind of essential to our definition of life as we know it. And it's something that is true of the smallest, simplest single-celled organism up to the most complicated multicellular organism like a human. So with all of these conditions, the presence of organic molecules floating around in a polar medium like water, like liquid water, distinct from its environment, performing homeostasis, coupling energy producing to energy consuming reactions and replicating and perpetuating itself. This is starting to sound like a fairly complete definition of life. And yet, I'm sure many of you are sitting there thinking, well, I can think of plenty of examples of things that aren't really alive and yet fit all of these criteria. A virus, since viruses are on all of our minds and influencing our lives quite a bit lately. Is a virus alive? It's contained and separate from its environment. It's replicating itself, but is it truly performing a metabolism? Does that matter? If we found a virus, discovered a virus on another planet, would you call that a discovery of life or not? Yes, I think so. Similarly, a lot of the processes we've talked about, can occur inside of a beaker of chemicals sitting on a lab bench. It occurs in a fire. It occurs at the bottom of the ocean and hydrothermal vents. And I'm sure you can think of plenty of other examples, prions, RNA molecules, DNA molecules yourselves. But the point here is that even all of with all of our criterion, our definition is still a little bit broad. And that's why we sort of go back to this answer. Well, you'll know it when you see it. What is it that makes us feel that a fire or a beaker of chemicals isn't alive, but an egg is? And so ultimately, I feel like some of the best definitions for life come from perhaps not the listing of criterion, but from something a little bit more personal, a little bit more innate, such as poetry. And so my definition of life, I wrote because I was challenged in a class that I audited, taught by a friend of mine, who's now a postdoctoral scholar at the University of Washington, Seattle. He challenged those in the class to come up with a definition for life. And the best I could come up with was this: life is that which tries. Clouds and crystals may grow and evolve. Stars may feed and die. A fire may spread its influence, but life is that which tries to survive, to grow, to learn, to adapt, to teach, and to be remembered in some way when it is gone. In other words, you'll know it when you see it. And so the next step in our journey, in our story, is now that we have a definition of life that is more or less universally unsatisfying to everybody, now we move to the problem of, well, how do we look for it? We're pretty sure that we know what isn't life, even if defining the full range of things that are life is difficult and fraught. But we have to start somewhere. And so with the definitions that we've got, how would a scientist go about looking for life in the universe? And the answer to that question is different depending on what scientist you are and what field, what perspective you're coming from. So if you're an astronomer, generally looking for life in the universe means looking up, looking out away from just the Earth, and perhaps examining the surfaces and the atmospheres of other planets for evidence of life or something close to life on another world. So let's see if I can get this cartoon going. So imagine you've got a planet. You've got a rocky planet, you've got water at the surface, you've got interesting chemistry going on in the oceans, and you've got the development of membranes and cell walls. You have something that's very close to the definition of life that we've established. How might you go about, if you couldn't see it directly and put your finger on it and hold a microscope up to it, how would you go about detecting it? And so the approach that astronomers have is to think about, well, what are the products of life? What are the kinds of molecules that might be metabolite replicating and maintaining itself? And could those molecules exist independently of the advent of life? Or do you need something like life actively producing them in order to detect them at significant levels at the surface or in the atmosphere of a planet? And there's a few molecules that are that astronomers tend to look for in the atmospheres of worlds that they think might be habitable, and that's oxygen, which we're all very familiar with. We need it to live and breathe. As well as things like methane and ethane, so simple organic molecules that might be the result of chemolithotrophic microbial life, or the result of the heterotrophic, the breakdown of complex organic matter into increasingly simple molecules. There's a wide range of metabolic products that might make their way into the atmosphere of a planet. But it's sort of the combination of different of these what's the word I'm looking for? The combination of certain metabolic byproducts in the atmosphere that astronomers might consider a telltale signature of life. The reason being that once these metabolic products enter the atmosphere, they can still react with one another. And there are certain metabolic byproducts that are unstable, that easily decay away or easily react away with other components in the atmosphere. And so if you're an astronomer and you're pointing your telescope at another planet and you're seeing the light either passing through the atmosphere of that planet, reflecting off of different layers of the atmosphere of that planet, you're getting this information about the interaction of light and the composition, interaction of light with an atmosphere of a certain composition. If you see certain molecules together in the atmosphere coexisting at detectable levels that really should be reacting away and decaying to nothing, then that tells you that there's some process going on at the planet's surface or even in situ in its atmosphere that is regenerating those molecules. So it's generally considered unlikely that you just happen to look at the atmosphere at the one moment that all of those molecules were first produced and sort of farted up into the atmosphere all at once. It's much more likely that you're looking at this sort of steady state, this time integration of processes going on at the surface. So let me slide forward a little bit. So your discovery process of a planet with this specific combination of metabolic byproducts in its atmosphere might involve a telescope, such as that at the Palomar Observatory. It might involve a space telescope, a satellite, basically looking out into the sky and collecting as much light as possible coming off of certain targets. And those targets are rarely the planets themselves, because the planets around other stars are incredibly small, and any electromagnetic radiation, any light that they might emit on their own tends to be, in most cases, dwarfed by the light coming from their star. So the astronomer is collecting light generally from the star and then looking at how that star's light might change in a periodic way that might only be explained by the presence of a body moving in front of or behind or approaching just the edges or the limbs of the disk of that star in the sky. Because as the planet is moving around the star, it's a little bit of the light of the star is passing through the sort of annulus of the atmosphere around that planet. Or if the planet's moving back behind the star, that tiny, tiny bit of light that might otherwise be sort of drowned out in the signal of the star, that tiny bit of light emitted by the planet or reflected from the planet, slightly shifted wavelengths, that's going away. That's disappearing in a periodic way as well. So an astronomer observing this distant star looks for these periodic changes in the spectrum. So that's the intensity of light at a range of different wavelengths or a range of different colors of the star. Looking for those periodic changes that might suggest the presence of a planet, and that might furthermore suggest the presence of a planet with a certain atmospheric composition. So the spectrum of the star, for example, when the planet's atmosphere is not sort of imprinted on the star's spectrum, will look different from the spectrum when the planet is imprinted from the star's atmosphere. And I've always sort of conceptualized these spectra as having a bite taken out of them. Like the planet's atmosphere is absorbing some of the light of specific colors from the star and maybe re-emitting it at a slightly different wavelength, or perhaps that signal is just lost. And when you get a spectrum, you can look at that specific pattern of bites, those absorptions taken out of the spectrum, and figure out from there what the composition of the atmosphere of that planet is. So there's a lot of limitations to this method of detecting life in the universe. One of the limitations being you got to have a extremely well-characterized stellar spectrum. You have to observe it for an extremely long amount, for just an extremely protracted amount of time to figure out those patterns of periodicity and distinguish true periodic changes in the star's spectrum from the noise and observational artifacts. And then even if you discount all of these analytical challenges, there's still the fact that, well, what you're ultimately observing is just metabolic products. You're not directly looking at an organism. You're looking at this broad time integration, this sort of steady state that the planet is in. So it's possible that you're missing a lot of information. Maybe there's life at the surface of a planet that's not producing metabolic products that interact with the light from the star at the specific wavelengths that you're capable of observing at a certain level of sensitivity from afar. You're missing information. Or it's also possible that because you don't necessarily have other information or other information or parameters about the surface properties of the planet, it's hard to say if what you're looking at is the direct result of true metabolism or life, or if it's the result of some sort of interesting photochemistry that doesn't really involve anything that we would recognize as a living thing. But it's a start. And so many astronomers are engaged in the task of figuring out, how do you distinguish a non-living planetary atmosphere from a living planetary atmosphere? What kinds of stars are going to be the best targets for these kinds of studies? There's a lot to learn even with those limitations. But astronomy is just one way of approaching these problems. So the way that I and many of my colleagues sort of approach it as planetary scientists who are interested in surface processes and subsurface processes is to turn to geochemistry, biogeochemistry. So tools that have been developed to study not necessarily distant planets around other stars, but the surfaces of planets and moons in our own solar system, and especially the geologic record of our own planet. So the approach of geochemists, the geologists, the biologists, the biogeochemists is to study not just whether or not there are those biosignature molecules in a planet's atmosphere, but to study the processes that produced them, to figure out how different elements, how different molecules might be cycled through the Earth's systems, as well as different types of living things. So all of the sort of the details of process that go into producing that steady state that an astronomer might observe from afar. That's what astrobiogeochemists are interested in the most. So this sort of cartoon diagram is one that many of my colleagues are familiar with seeing. Maybe y'all have seen them at some point yourselves. It's just a simplified model of the world, of planet's component systems. So there's land, there's bodies of water, there's atmospheric and weather systems, there's a temperature gradient through the planet's crust coming up. There are some rules of planetary evolution that we can assume to be true for many, not just assume, but that we observe to be true for many other rocky bodies in our own solar system and beyond. So with the simplified picture, one might imagine how a certain element, say sulfur, might travel between these different environments, how you might have some rocks with sulfur in them that when those rocks are heated in the Earth's interior, may be released from volcanoes and deep sea vents entering the atmosphere. And the sulfur in the atmosphere may interact with other elements while it's in the atmosphere. It may take on different forms, form different molecules of varying levels of size and complexity. And then those molecules might remain airborne, or they might be rained out over land or over the oceans, re-entering the sediment, the dirt, flowing down a river towards the larger bodies of water, and along the way, participating in all kinds of chemical reactions, being incorporated into different structures. And those reactions might be performed by living organisms, or they might occur spontaneously. Driven by processes entirely unrelated to life. And this cycle, as those sulfur-bearing molecules are buried and reincorporated into the rock, and these cycles occur over and over and over again, but they may occur at different rates throughout geologic time being sort of the time scales at which continents move, and as opposed to the time scale under which civilizations rise and fall. We're talking tens of millions of years. So in the course of a planet's history, the sort of balance of all these different processes, the nature of all these different cycles might change. And so a biogeochemist studies very carefully every step of every possible step that could be involved in the cycle, and every transition that, say, that sulfur atom might experience as it joins molecules of different sizes and different in different charges and things. And so geologists, chemists, biologists, it requires all of these different fields in order to characterize and understand all of these different processes, and the role of living things in them. But of course, that paints, even with this simplified model, kind of a complicated picture. So for the sake of just example, I'm going to do what most geochemists do, which is to simplify the world even further into the categories of atmosphere, ocean, sediment, and then deep burial. So if you sort of take everything that goes on at the surface and in the subsurface of a planet and simplify it down into those 4 boxes, and you simplify the transitions between those boxes into single arrows, you might get a diagram that looks something like this. So the arrows sort of represent a flux of a given material. Might be carbon or nitrogen or sulfur. It might be organically bound carbon or inorganically bound carbon. The model sort of works for all of these different chemicals. And so, for example, we'll say, going back to the sulfur example, say you had sulfate in the ocean. I'm gonna see if I can annotate here. So you have like a sulfate molecule. Ooh, bear with my, oh well. So say you have a sulfate molecule attached to something in the ocean. Say that sulfate gets thrown up into the atmosphere by a particularly energetic wave, by like salt spray or something like that. And perhaps that sulfate, while it's up there, bops around, and maybe it encounters some, maybe it ends up encountering some organic byproduct, something that's slightly more reduced than the sulfate molecule itself. And say it gets reduced a bit and it ends up becoming, let's say, hydrogen sulfide again. HS minus there. We're playing fast and loose with the oxidation state of the atom. That hydrogen sulfide might end up transitioning to sulfuric acid somewhere downstream, raining out, and say from a cloud as acid rain somewhere over a continent. It percolates down, becomes incorporated into the groundwater, or maybe it rains back out over the ocean and ends up going back into that reservoir of sulfate. So you can see you can start to imagine there's a variety of ways that any single atom might move between these different spheres, and how it might, let me stop the annotation, there we go, and how it might be cycled from between one box and another, and eventually make its way around. So let's see, I wonder if there's a way to erase annotations. Ah, brilliant. So still got this fairly complicated system. And thus far we've just talked about sort of the non-living elements of it. Imagine that there's boxes that represent living organisms in the ocean and the sediments, maybe even in the atmosphere. So each of these boxes, there's sort of a stable reservoir of elements in each of them. And there's these variable fluxes moving between them and in and out of them. And so a geochemist who is trying to characterize and understand how much movement of material there is between these different reservoirs, living and non-living, might assign a certain amount, describe the size of each of those reservoirs for a given point of time. And perhaps not just the size of those reservoirs, but some innate trait about them. And that innate trait, in the case of most geochemists, is their isotopic composition. So you have all these chemical elements, but most of the chemical elements have, of the lighter chemical elements, have multiple they have light versions and heavy versions of themselves, slightly more, slightly fewer neutrons, and that affects the stability of the bonds that they're capable of making in subtle ways, so that you might end up, again, integrating over a long period of time with a certain abundance of light versus heavy molecules in one reservoir or another. And so you can assign or you describe each reservoir not just by the quantity of a particular material in it, but by the isotopic composition of that reservoir. And bear with me, but this is particularly important because every one of these arrows moving between these different reservoirs, those involve not just the physical movement of material, but also chemical reactions. And because of those subtle differences in the stability of bonds involving more light elements versus more heavy elements, by passing through a particular chemical reaction pathway, you can end up biasing the isotopic composition of the final reservoir compared to the starting reservoir. So as your material is moving from one box to another box, you're getting these different sort of weights, average weights of the material in the different reservoirs. And so this is the really exciting part, is that if you can measure the isotopic composition of materials, of a material, say that sulfate or sulfide, or organosulfur compounds, if you can measure the isotopic composition of that form of sulfur-bearing chemical in any of these reservoirs, and based on theory and experimentation and just empirical evidence, say from field work or laboratory models, how large, how significant of a bias toward light or heavy any of those arrows causes. You can back calculate, you can figure out how much material was passing from one reservoir into another, and by what chemical pathways. So that's particularly exciting, because there are certain biases and the weight, the heaviness, the lightness of reservoirs that you would expect if there are certain metabolic reactions going on, certain chemical reactions that can only happen if you have a living organism there to couple the energy consuming to the energy producing reactions. And so by looking at the isotopic composition of these different reservoirs, you can figure out, was a particular metabolism in play, or was most of the flux of material from one reservoir to another facilitated by non-living reactions. So this is a little bit more detailed than the picture of the distinction between living and non-living that we were dealing with when we were just looking at a distant planet's atmosphere on its own. This is actually breaking down not just the living from the non-living, but the types of living. The metabolism of say sulfate reducing bacterium is very different than the metabolism of a photosynthesizing cyanobacteria, or the metabolism of a zebra. These all have their different signatures associated with them, that if you have a geologic record, so samples of records of the isotopic composition of atmosphere, ocean, sediment, deep burial materials, if you have that record to look at and measure, you can learn a lot about the history of life on a planet. So even with all of this complexity that we've walked through, this is still kind of a massive oversimplification. There are many more reservoirs than atmosphere, sediments, ocean, many thousands, and thousands of arrows between them. And so teasing them apart and solving those puzzles is the work of a planetary geochemist, or a planetary geobiologist. And so this currently, most of this kind of work is done with the geologic record here on Earth. So sort of treating early Earth in the distant past as if it were another planet. But as the types of analytical instruments that we're able to send to the surface of other planets continue to improve, these are the kinds of analyses that we can start to do for worlds like Mars, or perhaps worlds like Titan and Europa in the not so distant future. So the last way I'll sort of talk about the detection and study of life on a planet, and in a snapshot in time, as well as in the geologic record. The last and possibly most obvious form of life that we can think of finding would be a fossil. Uh, it's uh, it's all very well and good for scientists to talk about. Oh, we found X percent of methane in the atmosphere of this super Earth orbiting this M dwarf star somewhere else. Or it's all very well and good for geochemists to say, oh, well, we found this. We found this particularly light reservoir of reduced sulfur-bearing minerals associated with this particular sedimentary facies. That's all well and good, but if you're talking about a planet that's not Earth, where we know that there's been life for many millions of years, if you're talking about another world, then any one of those lines of evidence alone for the presence of life might not be enough, because it may be possible with environmental conditions on another world to sort of produce those signatures that are typically associated with life on Earth. You may be able to produce them abiotically on another world. So the astrobio community sometimes talks about what would constitute a smoking gun, something that's, okay, this is definitely a living thing created this signature on another planet. And the only single line of evidence that I could think of that would be that definitive would be an actual fossil, or short of course finding accidentally stepping on a gross bug or something when we send a manned mission to Mars. Short of that, finding a fossil, I think, would be very convincing. But fossils are probably the rarest and most difficult biosignature you could try to look for on another planet. It's hard to make a fossil. The likelihood of any individual organism entering the geologic record as a fossil is incredibly low. The fossil record is incredibly sparse and spotty. If, for example, you had a planet that was never inhabited by anything anything hardier than a jellyfish, you would probably be pretty unlikely to find very many fossils on that planet, even if that planet was just swarming with jellyfish, because it's so difficult to take an organism that is made up of this soft, easily digestible organic material and turn that into something that would withstand billions of years of geophysical evolution of a landscape. So there's a lot of steps that have to happen in order to take an organism from just dead living thing to fossil in order to discover it. And so some astrobiologists, including myself, this is a problem that we study in detail, because we want to know, okay, like how truly likely is it for us to find a fossil on this other world? And if we find a fossil in this particular context, what must that mean about the sequence of events that led to the formation of that rock? So I think I have another little cartoon up here. Imagine you have a single cell. Maybe this cell is in a deep subterranean aquifer somewhere on Mars. Maybe it's floating around in the sub-ice ocean of Enceladus. Wherever it is, you have a cell. It's performing metabolism. Different elements are passing through it in a variety of ways. It's doing its thing. But in order to enter the geologic record, in order for evidence of its presence to enter the geologic record, there's some specific things that have to happen, specific environmental processes. So that cell has to make it from the water column down into, say, the soft sediments. It has to be rapidly buried so that it isn't digested by other organisms and recycled and remineralized and put back into that complicated diagram we showed earlier. But if the organism can survive being eaten and can survive being advected out of the sediments by water flowing through them, then it has a chance, perhaps, of getting lithified. And this means getting replaced or even just encased by some sort of crystalline material, some sort of mineral. So if all of those stars align and all of those things happen in the right sequence, then it's possible that millions of years from then, a person or an autonomous planetary exploration tool like a rover might take a picture of what looks like a sort of maybe a mineral cast or a physical mold or some other sort of replacement structure that has similar dimensions and to the organism that once was. That would be a fossil. And it's not exactly a dinosaur femur. It's not exactly a great mammoth tusk or something like that, but a microbial fossil is really fascinating and exciting in its own right, because it tells you about, in many ways, it tells you about sort of the lowest, broadest, and most complex part of the world's ecology at the time that this rock was formed. It's those microorganisms that make up the bulk of the biomass on, well, certainly in the history of our own planet, and ended up shaping the environment around them. So that finding these microbial fossils is it's exciting. You have this thrill of discovery in a way that I personally feel that you don't necessarily get with the more obvious, more recent fossils of, say, vertebrates and those bony skeletons. So a microbial fossil, let me pause that for a moment. So that would be a fossil for an individual cell, where you can preserve in some sort of mineral that can withstand the test of time the form or shape of an individual cell. It's also possible to preserve structures that were built up by organisms, even though the individual organisms themselves haven't been preserved in minerals. And so this little cartoon here is just an example of, say, a microbial mat, just like pond scum on the bottom mud of a pond, sticking to some of the infalling sediments, facilitating the precipitation of certain minerals, and eventually building up a bunch of layers of sort of oddly arranged, maybe crinkly or wrinkly sediment that would be difficult to explain without the presence of life. And so the rock that formed from the lithification of those crinkly, wrinkly sediments can then be analyzed with a variety of techniques later on, closely imaged. You can measure the chemical and isotopic composition of the individual laminae, compare them to the chemical and isotopic composition of other structures nearby. And this, again, is a way of learning about an ancient ecology and figuring out its structure and learning something about how the living world was shaping the non-living world and for how long. So this is just a little cartoon. I stuck it in here from another talk that I did, where I was talking about some of the fossil microbial structures that I've worked with before, where even though I might not have individual fossils of individual cells that were part of the microbial mat, I can still see sort of this macro structure of wrinkly, sort of layered sedimentary rock. And I can analyze the chemical composition. So in this case, in the case of this image, the mineralogy of all of those distinctive layers. In the same way, at least conceptually, that an astronomer might look at the interaction of a star's light with the molecules in the atmosphere of a planet in orbit around that star. Instead, I'm looking at the interaction of a light with a known spectrum from the lab with the composition of this mineralogical sample. So by using the same sort of techniques of spectroscopy/spectrometry, I can determine what kinds of chemical processes facilitated by living and non-living things controlled the formation of this rock, or at least were happening at the same time that this rock was forming and lithifying. So, and this is just a photograph of a rock that is not from another planet, but is from a sort of 1.9 to 2 billion year old sedimentary package in sort of central Canada. If anyone is familiar with sort of the Thunder Bay, Ontario area, that's where this rock is from a formation called the Gunflint Chert. This is a really exquisite and beautiful example of microbially mediated structure preserved in the rock record for a very long period of time. So if this rock, if, for example, Curiosity, or if Curiosity took a picture of a very fine spatial scale, like picture of a rock on Mars, and found structures like this, that would be the scientific discovery of possibly the greatest scientific discovery since the beginning of scientific discoveries. That would be really cool, really definitive evidence of microbial life on another planetary surface. Because you can't make these structures, these sort of wiggly, lumpy structures, abiotically. And not just that, but the nature of these structures tells you something about not just the presence of life, but the style of life. It means that there were lush microbial mats, so sort of carpets of mixed assemblages of microbes with a variety of different metabolic styles, likely photosynthesis, as well as chemotrophy, as well as heterotrophy, coexisting within millimeters of each other and sticking to and accumulating carbonate chemical sediments at or just below the sea floor in this ancient ocean. Now, that's a lot of information and something that you really couldn't get without that close in situ analysis of mineralogical samples, and something that you really couldn't understand unless you were bringing in tools from chemistry, tools from microbiology, tools from geology and petrology and just plain geography. Like, you need all of these different fields in order to really understand and read this record of ancient life. So, moving on from there, the question that we set out to answer at the beginning of this expansive journey was, what is life? And once we've decided that we have a sufficient definition of life, how would we go about looking for it on another planet? What would it look like? And how would we go from just identifying the presence of life to actually understanding what it was doing and how it was influencing and participating in feedbacks with it, with the non-living elements of its environment. And so ultimately, future researchers who go to the surface of other planets or go to sit in orbit around a world, around another star, who knows, those researchers will probably end up using basically the same tools that we use to study ancient Earth history, that we use to look back at the history of life in deep time on our own planet, as well as the tools of astronomers working with Earth-based and space telescopes and other remote sensing techniques. So with that, I am going to go ahead and end the presentation. Just put up again, my name is Cecilia Sanders. I study geobiology. I study the coevolution of planets and living things on planetary surfaces. Ultimately, my specialty is wiggly things on rocks, wiggly things in rocks, wiggly things around rocks. Do feel free to visit my website and drop me a line if you think of a question later that you are very excited to pursue further. I'm always happy to talk about this subject, but I'll go ahead and stop there. And I think I was told we maybe have a half hour or so left for Q&A, depending on how sleepy people are feeling.
Yeah, if I could jump in and ask the first question. By the way, this is a very thought-provoking talk for us. I gather from what you said earlier that maybe Europa is not the best place to look for life, that we ought to be doing like what they're doing with Jezero and looking at the deltas that might have these stacked up sediments and so forth. Where would you go first if you had the call?
Yeah, so if money and time were absolutely no object, certainly I think it would be incredibly valuable to study every object in the solar system, because I think that even worlds where we're unlikely to find life, unlikely to find things that we recognize as life, we would still learn a great deal about prebiotic chemistry and the chemistry of planet formation and evolution, because understanding why life is where it is and why it is the way that it is requires us to understand why it isn't in some places, and say the differences between the worlds that do host life and the worlds that don't host life. So I think it would be incredibly valuable to study any body in the solar system as minutely as we've studied the record of deep time on Earth. That would be incredibly informative for the study of the origins and evolutions of life. But where I personally might want to go, I think would be it's like cliche, but I think Mars is just a really fascinating, beautiful world. It's similar to Earth in so many ways, but it has, because it's had sort of it's smaller than the Earth. It has never been as tectonically active as the Earth. Its insides likely cooled off earlier than the Earth's did, and for all of these reasons, the rocks that are at the surface of Mars are much older than most of the rocks at the surface of Earth, and so preserve this a much more ancient record than we have on Earth of what was going on in the early solar system, sort of with the early development of planetary oceans and planetary atmospheres and stuff. So I think I would be curious to look closely at not just the surface, but also like the shallow subsurface of Mars, and And again, if money were really no object, then maybe sending some kind of drill probe that would be able to maybe make it down below the sort of permafrost line. Because even though Mars isn't that tectonically active, it still does have a geotherm. That means it still does, it's hotter the deeper down you go compared to the surface. And that means that you can have liquid water. It's either groundwater or true like aquifers and things. I think that would be a really interesting place to look for evidence of life or examples of perhaps early prebiotic Earth. Europa, I think outer solar system stuff. Europa, Titan, Enceladus, Io, even these places are really wild and out there, and I think are probably less interesting targets for the study of for like the detection of life, and more interesting targets for just the study of the diversity of planetary evolution. And life is a part of that, certainly, but if I'm going to try to figure out was there life here or not, those probably wouldn't be the first picks. But that said, this is all based on the assumption that we're pretty sure we know what life is and what its parameters are. So maybe there are tentacle monsters on Europa. Maybe we shouldn't discount that offhand. Let's see. So looking at the chat, someone has asked whether or not viruses are living. I guess that was a little earlier on in the talk, but yeah, I mentioned viruses briefly because viruses sort of require, being a virus is all about kind of hijacking the cellular machinery of your host so that you can perform, so using the cellular machinery of your host to perform the chemical reactions necessary to replicate and perpetuate yourself. So a virus on its own doesn't have the internal machinery to perform metabolism, to couple energy consuming to energy producing reactions. It requires the transcription, translation, machinery. And I keep saying machinery. What I really mean is diversity of enzymes and nucleic acids that the host cell has in order to survive. So a virus, many biologists don't really countenance viruses as being like true living organisms. However, the existence of a virus sort of inherently implies the existence of other organisms that do have that cellular machinery that can allow that virus to perpetuate itself. So if you, I forget the exact numbers, but if you take a liter of seawater and were to measure the abundance of cells versus the abundance of viruses in that liter of seawater, I think viruses you'd have, I'm going to get the order of magnitude wrong, but I think it's something like 100 to 1,000 times like the number of individual viruses in that liter than bacteria and protists and things like that. So it's viruses are everywhere on Earth, and the reason that they're everywhere on Earth is because there are living organisms that have that complex cellular machinery that viruses can take advantage of. So it's so whether or not viruses are living, if you found them on another planet, then that's like, whoa, that's its own smoking gun, regardless of whether or not we consider the virus alive semantically or not. There's let's see, someone mentioned the Miller-Urey experiment. So at the beginning of the talk, I talked a lot about the primordial soup, this sort of the transition from a non-living world to a living world, which required the presence of water in contact with rock and sediment, and the sort of evolution of increasingly complex molecules that resulted from that environment. The Miller-Urey experiments are a famous historical set of experiments where there was an attempt to sort of replicate prebiotic chemistry to, if you recreated in a bulb, in a closed bulb in a lab, the kind of environment that you might have expected on early Earth. So little to no oxygen in the atmosphere, water with certain kinds of solutes dissolved in it, the presence of energy sources such as heat from the geotherm or even storms and lightning in the atmosphere. If you put all these things together, Miller and Urey wanted to know, could you produce some of the essential precursors to life? And not only can you go from those essential precursors to something that we might recognize as a cell, or something that we might recognize just as metabolism. And so there have been many recreations of the Miller-Urey experiments with increasing levels of complexity and with increasingly better constraints on what the actual conditions of the early Earth were. And that is a really interesting rabbit hole of research to go down if you're curious. Yes, farted up. Sorry I feel like some of my professional speaking skills have atrophied a little bit in the wake of the pandemic. And so, excretion is just like, somehow to me seems like a nastier and more inappropriate word than just farts. But metabolic byproducts, farts. Let's see. Any thoughts about the detection of phosphine in the upper atmosphere of Venus? That was really exciting news when it came out earlier. For those who maybe missed some of the controversy surrounding that drop of new research. Venus is much closer to Earth in size than Mars, but it is incredibly different in its surface conditions and its history of planetary evolution. Venus is hot, Venus is hazy, Venus is an incredibly inhospitable place to life as we're familiar with it here on Earth. And so, and many attempts to study surface processes on Venus have been thwarted by these inhospitable conditions at the surface and in much of the atmosphere. So the detection of phosphine, which on Earth is exists as a metabolic byproduct, so the result of living things going about their business. The detection of phosphine in the Venusian atmosphere was this very exciting thing. Like, oh my gosh, like, does that mean that there's life on Venus creating this phosphine? But I am by no means expert or well-versed in this particular area of atmospheric research on Venus. But my understanding, when I read that paper and some of the responses to that paper from the community, my understanding is that it is possible to create phosphine abiotically under conditions with extreme temperatures and extreme acidity, which are the hallmarks of the Venusian atmosphere. So it's possible. So my understanding was that while it's possible that phosphine in the Venusian atmosphere could be a biosignature, while that's possible, what it definitely is evidence of some really interesting really interesting atmospheric chemistry going on. So regardless of whether life is involved, there's a process going on in the Venusian atmosphere that we're just now starting to probe and understand using remote sensing techniques. Then there's the question, what would surprise you most if life was discovered, say if something didn't fit your model? I think it would be very surprising and very interesting to see something performing kind of the functions of life as we've discussed it. So metabolism, self-replication, all of those things, performing it using a different set of fundamental biomolecules or building blocks. So the way back at the beginning of the talk, I was talking about how organic molecules, so molecules with sort of a backbone, a structure of carbon, oxygen, and hydrogen, organic molecules are fairly special because the bonding environment that carbon, hydrogen, oxygen are capable of allows them to create incredibly complex structures and a wide diversity of molecular forms. And for that reason, it's possible to, and for that reason, with organic molecules, it's possible to create specialized molecules that are very good at certain things. So nucleic acids, storing information, storing complex information, and being able to transcribe that information into protein structures. Proteins, protein structures are incredibly complicated. They can be so specific as to only admit one very particular type of molecule with one very particular in one very particular oxidation state inside of a cell, or being able to force reactions that would never spontaneously occur in nature, to force them to occur by bringing molecules close together that in orientations that are highly energetically unfavorable. Like organic molecules are amazing at those things, but the organic molecules that form the basis of life on Earth, they are not the only organic molecules, capable of performing those functions. So there's I think it would be very cool to see something doing like the work of an enzymatic protein, or doing the work of a nucleic acid, doing the work of a fatty acid or something, but having fundamentally different chemistry, fundamentally different chemical composition than the biomolecules that we're familiar with. I think that would be really interesting and cool, and possibly sort of paradigm exploding to see that happening, because theoretically we're like, well, we use the biomolecules that we've got because those ended up winning the evolutionary race early on enough that they just became what we use. So I think finding that different basis in another world, or perhaps even in some isolated niche on our own world, I think could be very revealing.
Thank you.
Let me ask another question. If we expect to find active living organisms of some kind or another, say they're small, is there a moth to the flame experiment you could do? I would think sugar, but a good friend of mine who's a medical doctor insists that every living organism desires alcohol. And he said, even at the cellular level, you put a little bit of alcohol and they come running, so to speak. I'm not suggesting beer on Saturday afternoon football games, but is there anything that you could that you could attract them with, that you'd
Oh.
have some degree of certainty that it would work.
I love that as an idea. I've never heard that, really. The idea that you could, you know, bait a trap for a living thing, but in some ways, like that does make sense to me. So to some extent. So for example, so using oxygen as an electron acceptor for our metabolism at the most fundamental level. Like humans, we're heterotrophs. We consume organic matter, we oxidize it, and that oxidation of the organic matter, that's the energy producing reactions that we can use to couple to all of the energy consuming reactions necessary to build and maintain our bodies. So oxygen is an extremely good electron acceptor. It loves electrons. It's extremely easy to oxidize things with oxygen. That's why we made the term oxidation. But for the same reason, so even though oxygen is such an efficient way to carry out these fundamental catabolic reactions, life on Earth had to evolve the capacity to use it. And indeed, there are a number of life forms on Earth, and indeed the bulk of life for most of human history, for which oxygen was toxic, because for the same reason that it's such an effective electron acceptor in the course of our own metabolisms. It can kill many organisms. And so it's interesting because there are, just because a particular molecule is really useful in some way, really useful in like a sort of basic chemistry, theoretical chemistry way, like, oh yeah, you can get a lot of energy out of this reaction, or oh yeah, this reaction happens really quickly, so you should expect it to win out over others. Like, just because a molecule is really good at that in some circumstances doesn't necessarily mean that a living thing is going to have the right cellular machinery to use it, to its advantage. So I think that, like, I feel like one would be hard pressed to find like to find something that you could present to any living organism sort of ubiquitously, and have that living organism want to and be capable of using it. I think you'd be hard pressed to find like a particular substance like that. But you could look for the presence of specific metabolisms in action by presenting sort of, a range of different treats. For example, one of my colleagues, Usha Lingappa, who's now, she got her PhD at Caltech in geobiology. Now she's a postdoctoral researcher at UC Berkeley. Super brilliant, innovative scientist. And she did this awesome experiment where she took sunlight, just ambient sunlight, and had it shine through a prism, so that white light could be split into a nice little rainbow spectrum, so spatially distinct colors of light, and then shine those on a microbial mat. A microbial mat sample from one of the places where we did some of our field work. It's actually in the Turks and Caicos, which is a really cool trip. But so she shined this light, all these different colors, onto the microbial mat, and then sampled for DNA, RNA, and proteins in each of those segments that were exposed to those different colors, those different wavelengths of light, to see how you could to sort of see which taxa were enriched and which taxa were depleted when you provided light of only this certain wavelength, because the microbial mat as a whole has like tons of microbial diversity in it. But there are certain wavelengths of light that certain organisms were much more effective at utilizing to drive their metabolisms than others. And so, you know, that's just like a small example, but basically it was, let me take this community and let me learn not just about who all is there, but learn about let me learn about how they function. Let me learn about their internal cellular machinery for photosynthesis by providing them with different conditions, different environmental conditions. And so you could, you know, maybe you go set up such an experiment on the regolith of another planet, or see if you can induce certain environmental changes or promote the growth of certain organisms over other organisms by providing them with these different sorry for the background noise, my dog is doing her nightly run through the house. But yeah, like I could envision an experiment like that where, you know, if not like some universal like carrots to tempt all living things of all stripes out of the woodwork, you could certainly test for specific styles of life in different environments. Yeah, I see there's also one more question over here in the chat. Because of the sheer size of the universe, is the search for intelligence at or exceeding human intelligence a futile search? Excellent question. I did not talk about the search for intelligent life at all in the course of this talk. I've mostly been focusing on microbes because I think, you know, microorganisms are the, you know, they're the base to everything. They're the first kinds of life that emerge and the ones that are, you know, dominant and ubiquitous. But the search for intelligent life is certainly very captivating to the human spirit. And I think the trouble with the search for intelligence beyond the solar system is because it relies on searching for signals sent out by like an intelligent civilization. And, you know, that by its very nature is just assuming so much about civilization and, you know, the definition of intelligence, the nature of intelligence. You know, I think that you know, there's not really a scientific basis to the idea that single-celled life will always develop or evolve toward increasing complexity, and eventually that intelligence that like begets intelligence, and that an intelligent species will build a civilization that invents the television and the radio and space telescopes. I think that there are myriad examples of human history of societies that didn't go through that sort of regimented technology tree because of the resources that were available to them, the geography, the geography that they occupied, and just because of sort of the diversity of human values. And, you know, if we have that variation amongst the intelligent species as we see ourselves on our own planet, I think it's difficult to, I don't think it's really possible to like prescribe any particular style of technological development to another civilization. And I've never quite, and you know, it's not my field of research to understand the, I'm not a cosmologist, and I don't really know if there's scientific basis to the idea that just because, you know, in an infinite universe, everything has to happen somewhere. I don't really know if that's true. Slash, I don't know how one would prove if it was true, which is why it's sort of the realm of cosmologists and theorists to think about. But yeah, I personally think that the search for intelligence in the universe, as long as it relies on a lot of these sort of anthropomorphic and honestly industrialized, like assumptions about industrialized societies, I think that it will be a futile search, because I think that there's no reason that intelligence has to mean sending out signals into space and building spacecraft. I think that but that is to say, I don't think that means that there isn't intelligent life out there, or that it couldn't be found necessarily. But I think that just like how we have to develop kind of a simultaneously inclusive but specific definition for life as a whole, I think an inclusive but specific definition for intelligent species or civilization like also has to be developed to like sort of focus that search. But super interesting question. I think about it a lot. Any more questions?
No more questions. I'm sure we'll think of questions, because this is a very thought-provoking talk that you've given us, and it's not something that we hear regularly. This is very multidisciplinary in terms of our thinking, and you've really opened our eyes to lots of things that we don't normally consider.
Yeah, well, thank you for indulging me. This is one of my favorite things to muse about. And yeah, there's you know, we're at a moment in, I think, the history of science where, you know, there are very few questions left, unanswered questions left that can be answered, you know, just by chemistry or just by biology or just by astronomy. I think you really need all of those different fields, all those different styles of inquiry to get at these questions. And so, yeah, happy to talk about it ad nauseum.
No, it's just great, though. Thank you so much.
All right, thank you guys.
Is there anything we can do for you? Normally I ask people this, and if they're from JPL and they're taking measurements, oh yeah, somewhere down the road you can use your telescopes, but I'm not sure what we can do here.
Oh, well, I
This
might
is the science basis.
yeah, I might come out and join y'all for some of your various astronomy activities. I used to be in the undergraduate astronomical society when I was an astrophysics undergraduate at Harvard, and would run those dark sky trips, and they're just such a joy and always remind me, like, you know, the reasons why we got into these highly specific and niche fields of study in the first place was, you know, because stars are cool. It's cool to look at them.
Yeah.
So I might follow up with you just for the activity schedule, because that'd be really cool.
You're welcome anytime.
Awesome. Thank you guys so
Yeah, I'd love
much.
to have you at the site. Absolutely.
Wonderful. Thank you so much.
Thank you again.
All right. Have a nice night, everyone.
Okay, thanks for being with us, and the
Thanks,
great
Cecilia.
talk.
Thank you.